Hello, everyone. Welcome to the Bulkley Valley Research Center Seminar Series, Virtual Edition. My name's Dawn. I'm the Operations Manager at the Research Center. Thanks so much for joining us today. I'm really looking forward to hearing from Haley. I'd like to start by acknowledging that I'm hosting this seminar from the traditional territory of the Ginnamden clan of the Wet'suwet'en Nation. I'd like to also acknowledge our funders that help support the seminar series happening, Bulkley Valley Community Foundation and Wet'suwet'en Community Forest. They are really great partners for the center and allow us to do some things that wouldn't otherwise be possible on our small operating budget. And thank you to all of you for joining, for sharing your time and your interest, for purchasing memberships, and generally keeping us going. It's much appreciated. Thrilled to be hosting Haley. She's a research scientist with the Ministry of Forests, working on tree breeding and genetic conservation. Her work in conservation genetics has taken her across countries, systems, and species. She was just sharing that she's from the east coast of the United States, so very different environment than here. She's really enjoying her time in BC and has promised she's going to visit us in Smithers. She's been a postdoc with Sally Aitken at UBC examining landscape level genetic diversity, and she wants to use her work on genetic techniques and theory for the effective management and conservation of natural resources. Welcome, Haley. Thanks so much for joining us.
Thanks so much for having me. I am sorry that I could not be there in person, but I am speaking to you from the Cowichan Lake Research Station, which is here on Vancouver Island and is on the traditional territory of the Kwetsin and Subaset nations. And today, rather than sharing with you very specific research that I've done in the past, I'm going to give you a broader overview of how we manage genetic diversity in BC forests. And so rather than, again, just my research, this is policy, practice, and research that's been conducted over the past several decades. But we're going to go over why we should care about genetic diversity in commercial forestry, and also in conservation here in BC, and kind of think forward about what the future may hold. And so we're going to start off very general because I know that genetics can be frightening to people. I always say that someone in the past decided to make genetics seem scary to make sure they keep their job, but that is not what I'm going to do today. We're going to start off with the basics so that whether you had a genetics course last year or 30 years ago, we should all be on the same page. And so we all know that genes are what code for our physical appearance or our phenotype. And so sometimes this is very simplistic and straightforward, where you have one gene, so one sequence in your DNA or genome that's coding for a phenotype, so something like eye color. But many times this is more complex, especially when we're looking at more complex quantitative traits, something like height. In this case, it's normally polygenic. It's normally that you have many different genes that are coding for this, what we consider a single physical trait or phenotype. And so since we all have different physical traits, we know that each human also has their own unique DNA. But what we often don't think about is that plants also have DNA. And again, they can have very simplistic traits where one gene codes for them. So something like flower color, sometimes insect resistance is what we call major gene coded. So there's one to very few genes that are coding for it. But oftentimes when we're looking at these physical phenotypes, there are many genes that are coding for it. It's much more complex. So something like drought resistance would be highly polygenic. It would have many different genes coding for it. And when we're not talking about clonal plants, when we're thinking about most of the tree species that we see here in BC, each individual tree also has a unique genotype or genetic DNA. And we also know that when we're looking across different species, looking at species diversity across the province, we know that different species exist within different environments and climates. So something from coastal Douglas fir existing here on the coast to lodgepole pine in the interior to something like white bark pine, which is up in those alpine ecosystems. And we know that these different species have different adaptations that allow them to live in these different environments. And that underlying these different adaptations, because these are physical appearances, are again different genes. These would have quite different genetics. But what we don't often think about is the fact that there's also diversity within a species. you can look across the full range of something like coastal Douglas fir and see that there are, in fact, different climates and environments that the species is adapted to. And so this is something that we in forestry study a lot through what we call provenance trials or common garden trials, where you would take individuals from different provenances or populations across a species range and grow them all in one location, that common garden. And so in this example, we see Sitka spruce that was collected from across its range all the way from California up to Alaska and grown at UBC. So photo credit and experimental credit to Sally Aiken's lab. And so when we're looking at when we're looking at the differences across a species within a single location, this normally means that they have a genetic basis. And so this local adaptation usually means that there's some genetic link. And so when we're looking within a species, we also know that there's genetic variation. And so that would mean that if we have different genes that are coding for different physical attributes, the more genetic variation you have, the more variation in traits you have. And so one idea as we're moving forward into climate change would be that you would potentially target the specific gene that's coding for that trait of interest. So let's say that you find the genes that are underlying drought resistance, you could then target more of those genetic variants that code for more drought resistance when you're planning populations. But we can see how this pans out. And so let's say we have two different populations, one where we have a lot of those drought resistance genes, and one where we just have a variety of many different genetic variants. And so when you have something like drought coming through, something that's quite predictable as we're moving into climate change, you would see that that population that has more drought resistance genes would do better. The problem with climate change and with just the environment in general is that it's quite stochastic and unpredictable. Climate change also has a lot of extreme events that are harder to predict, random frosts, random heat waves, as well as side effects like increase in insect outbreaks and pathogens that we can't always predict. And so in this case, this is where you see that the population that has more genetic variants that might not do better when we're looking at one stressor can do better in reacting to many different unpredictable stressors. Which is why greater genetic variation usually means that you have a more resilient population. And this is because genetic diversity is that raw material for natural selection. So genetic diversity is really what underlies adaptation to a local environment. And so there's many different ways to measure genetic diversity, but we'll just talk about two today. And so the first is heterozygosity and is fairly straightforward. So we know that when we're looking at when we're looking at a gene, it would have two different potential gene variants at that location. So you get one allele or variant from the maternal and one allele or variant from the paternal. And so a heterozygote is when they're different, big A, little a, and a homozygote is when they're the same. And so heterozygosity is just looking at the proportion of heterozygotes, whether it's within an individual or what we're talking about here across the population. And they have found that heterozygosity is well correlated to fitness. And so usually the greater heterozygosity you have, the more fit an individual or population is. Now, the other term I'm going to talk about, which will come up later, is effective population size, or NE. And so this one is much more complex. And rather than census size, looking at just the raw number of individuals that are in a population, it's instead looking more at the individuals that are contributing to the next generation. And so a common definition for NE is the number of individuals genetically contributing to that next generation. But this is actually incorrect. It'd be nice if it were that straightforward, but it is actually the number of individuals in an idealized population experiencing the same rate of genetic drift. Now, we are not going to get into what genetic drift is. I could teach, and well, maybe not me, but someone could teach an entire semester on effective population size alone. It's an incredibly complex concept that even population geneticists struggle with. But if you're looking at a population and you have totally genetically unique individuals that are not related whatsoever, and they produce the exact same number of offspring, then that would be equivalent to your NE. So let's say you're looking at this population here, which may actually have 9 individuals, but for the census size, but your NE would be 4. And so NE is impacted by things like the number of parents, the relatedness of parents, and the evenness of parental contributions. Now, when we're actually looking at these measures from a population. It's quite straightforward. Bigger numbers equal more genetic diversity. So heterozygosity is proportion, so it varies from 0 to 1, whereas effective population size can vary from anywhere from 2 to infinity. And heterozygosity is more important for adaptation in the short term. So it is what natural selection is directly acting upon, whereas effective population size is more important for ensuring genetic diversity across generations. And these two are actually related, which shows you how effective population size looks across generations, can actually predict the loss of heterozygosity as you're moving forward in time. So you can see here that a higher effective population size would mean a slower loss of heterozygosity over time. And when we're talking about natural populations of trees, especially those that are large, continuously distributed populations like most of our conifer species here, they are actually highly genetically diverse. And this is down to the fact that they are large in population size and are wind dispersed. And so they actually have more naturally existing levels of genetic diversity than most other living organisms, much more than most animal species. And this wind dispersal, which means that it can spread pollen and therefore genes up to tens to hundreds or even thousands of kilometers, also means that different populations are highly genetically similar. And so you can see this graph here on the right, which is from a paper by Maloney et al. From 2019, which was looking at the distribution of warm alleles and I believe lodgepole pine. And so this is actually looking at the frequency of alleles or gene variants that are associated with warmer climates. And so you can see that we're looking at frequency. And so none of these populations have zero. And so most populations have genetic variants that every other population have. They just differ in frequency. There are exceptions to this, species that are highly fragmented or have different dispersal vectors than wind, or are in decline due to factors like habitat destruction or pests or pathogens. And white bark pine checks all of these boxes, and we will talk about this later. So now we'll get into how we are managing genetic diversity in BC forestry. And so to get into this, we'll just go very briefly over what tree breeding actually does. And so the very first step in tree breeding is to go out to a natural population that we know should have really high genetic diversity and select trees that have the desirable or usually valuable physical traits. So easiest to think about this is height, but there could be other traits like diameter at breast height. So they would select those species, and these would then be used as parents to breed and then look at their progeny or their offspring, and then again select those individuals that have those valuable traits to again serve as parents in breeding and produce offspring, and again select those trees that have those valuable traits. And so it's moving forward in generations, breeding, and just continuing to select for those trees that have the traits that we desire. Remembering that at each step, each one of these, what we call generations or cycles, you're producing seed that is then used in reforestation. And so here we're forming what we call the breeding population, but this what actually goes out onto the landscape for reforestation is what we call the production population. And so this is done using seed orchards where you'd have those parents that you had selected for those desirable traits to be used in a seed orchard. And so what happens is we actually clonally replicate them, which is pretty cool, using grafting. And so you could look out at a seed orchard if you've ever visited one, you might see many hundreds of trees, but it would actually only be representing, say, 100 what we call genotypes, so 100 genetically unique individuals. And then each year, these seed orchards produce seed, what we call a seed lot, that bulking of all of the cones that those trees would produce from mating with one another. And this is what actually gets used in reforestation. And so the goal here is that these planted stands then, rather than looking like a natural stand that, yes, has a lot of genetic diversity, but is then also very physically diverse, we get to physical uniformity, which would also mean that it would be more uniform genetically. And this is what we call genetic gain, moving from that natural full set of physical diversity to a set of trees that are producing the most timber and have the most desirable traits. But as we saw earlier when we were looking at those two populations, the one where we're aiming for just a specific gene variant, when you have a stressor, something like an insect infestation, the populations that have lower genetic diversity are more vulnerable. And this is why tree breeding has always been a balance of genetic gain versus genetic diversity. And genetic diversity has always been considered in tree breeding. So if you have too much genetic gain and too little genetic diversity, your population is then more vulnerable to these different stressors, whether it's climate change or insect or pathogen outbreaks. But if you have high genetic diversity and virtually no genetic gain, then you're also losing out on timber production. And so this not only means that you would have to harvest more and reforest more of the land to get the same amount of timber, but then we're also losing out on the economic value of these stands, which is incredibly important. We have communities and First Nation communities that depend on forestry here in the province, and a lot of the revenue that's produced by forestry goes back into social services like healthcare and schools. And we all live in houses that are mostly made out of wood. And so this is something that we do need to balance in order to have successful forestry. And genetic diversity is managed at three different levels of forestry. So you have that breeding population, so all of those parents that were pulled first from the natural population and then from the offspring that are produced in breeding, forming your breeding population. And then you have that production population formed by the seed orchards and the seed lots that it produces each year that are actually used in reforestation. And then finally, what actually goes out on the landscape in those planted stands. And so policy here in BC actually manages genetic diversity at this seed lot level. And they've set the policy around effective population size, and it's required that every seed lot that is actually used operationally, that is actually getting planted out for reforestation, must have a minimum effective population size of 10. And this number was not pulled out of a hat. So this took a lot of research. So Alvin Yanchuk and John Russell were the two that were behind most of this research. And if you are interested in this and want to see their full reasoning, this is available online. You can search deployment issues in genetic diversity and read their full report. But what they looked at was how heterozygosity would decrease over different generations based on different effective population sizes. So based on that original relationship that I talked about previously. And so here we're looking at the loss in heterozygosity from the natural population. So looking at that full set of genetic diversity across generations, across different effective population sizes. So going from 6 in that red all the way up to 1,000 in that purple. And so you can see that the lower effective population sizes have much more rapid declines in heterozygosity. But what we're talking about here is seed that's being used for reforestation. So this is seed that's being planted to eventually be harvested, usually at around 50 years. Which means that we're really only interested in that first generation. That's all the time the seed is going to experience on the landscape before it's harvested. And so we can zoom in on that first generation and look at the capture of total population heterozygosity, again, comparing this to a natural population that's highly genetically diverse, and compare different effective population sizes now along the x-axis, just zooming in on that first generation. And so we can see that in any of 10, our policy minimum preserves 95% of the original genetic variation after one generation. And so this is not 100%, but again, we're aiming for that balance between gain and diversity. And it's important to remember this is also a seed lot. So this is just the seed that's being produced in a single seed orchard, for which most species have multiple seed orchards when we're deploying across the province. And not that breeding population, all of the potential parents that could be used for deployment. That's the breeding population. And while there's not set policy around our breeding population, minimum effective population size, all of our breeders target an NE of 80. And so we're getting much closer to that 100% of genetic diversity. So you might be wondering why we would have picked effective population size as the metric to base genetic diversity, considering it is an incredibly complex metric, and why would we not pick heterozygosity? This is because heterozygosity really can only be measured using genetic data, whereas effective population size, because it has that relationship with the census size and reproduction, we can actually measure on the ground. And so every year, for every seed lot that is produced, our orchard managers do estimate an effective population size because we know that reproduction in parents and even in those clones or ramets within parents can vary. And so they will go out and determine which trees are producing pollen in cones in order to estimate that parent contribution and determine how many parents are actually reproducing. But this boots on the ground measure is based on observation. And so a next step to check to make sure that our genetic diversity policy is working the way we thought would be to compare this to estimates from genomic data, since it is based on genetic drift, something that's nearly impossible to measure without genetic data. And so this was a project that was done in collaboration between MOF and UBC from Genome BC funding, where we compared that boots on the ground estimate of effective population size, which is shown here in orange, to two different estimates of NE from genomic data, so just using two different methods. And we did this for 12 orchards from lodgepole pine. And so you can see here we have our genomic estimates are bounded by 95% confidence intervals. And for those of us who haven't taken a stats class in a long time, that just means that if your measure is within those 95% confidence intervals, when you're comparing them, they're not significantly different. And so we have our policy minimum NE of 10 and the dashed line down here on the bottom. So first thing to note is that all of our estimates are above NE of 10, which is great. It means that we're at least definitely meeting our policy. But the other thing to look at is that our orange measures, the metrics that are currently being used to monitor genetic diversity, are oftentimes below our genomic estimates. So looking at orchard 2, 4, and 5, you can see that they're quite underestimating based on our genomic estimates, which is actually good news in terms of genetic diversity. An underestimation would mean that there's actually more genetic diversity in reforestation than we would think based on our current estimates. But when we're looking at that graph, so now we know that we're hitting at least NE of 10 when we're doing our monitoring. It would be good to also make sure that we are actually capturing 95% of the heterozygosity that is found in natural populations. So it also makes sense to compare genetic diversity in seed collected from natural stands versus seed collected from seed orchards, which is also what we did as part of this project. And so again, looking at lodgepole pine, we're comparing 12 of those seed lots, so seed collected from seed orchards, to seed collected from 33 different natural stands. So the natural stands are in green and our seed orchards are in blue. And this is showing heterozygosity, again, bounded by 95% confidence intervals. And so we can see that there's no significant differences between our natural seed and our seed orchard or the seed that's coming out of tree breeding, which means that we are doing a great job of capturing that 95% of heterozygosity. Now, where we do see differences start to emerge is when we compare effective population size estimated using genomic data between our natural and orchard seed. And so here, the natural seed has much higher effective population sizes, with most of them having upper confidence intervals or estimates of infinity, which means that there's virtually no genetic drift going on in these much larger natural populations. So this isn't really surprising. It is based on the number of parents that are contributing to a population. And so there is essentially infinite parents in a lot of these large, continuously distributed populations versus that subset of parents that would be used in breeding. But this isn't a huge concern because we do see that our heterozygosity is the same. And so a common question that we are getting is any of 10 enough genetic diversity? And so my response to this is usually enough genetic diversity for what? And so again, we're looking at reforestation here. And so we're looking at this balance of gain versus diversity, but we're also looking at seed that is destined to only be on the landscape for a single generation, in which case we are still capturing naturally occurring levels of genetic diversity. And so instead, we're also looking at whether any of seed lots is even the right question. Because again, we're managing genetic diversity at these three levels. And so we know that we have a target NE for the breeding population. We have policy around that production population, but what's actually happening, what's going on in the landscape, which is where genetic diversity would matter the most. And so as part of this Genome BC project, we also looked at this. Looking within coastal Douglas fir, we looked at effective population size in seed lots, and then also measured it in two planted stands planted with each of these seed lots. So we had two sets. So we had two different seed lots that were planted across two different stands. And our stands were sampled at around 15 years of age. So they would have been approaching maturity. So they would have already experienced some competition as well as some different stressors that may have caused mortality. And what we actually found when we looked at our seed lot NE in blue versus our stand NE here in purple matched up to the seed lots they were planted with is that our stand effective population size was actually higher than our seed lot NE, which doesn't make a lot of sense. How do you gain genetic diversity just by being planted in the ground? And so the most likely explanation for this is natural regeneration, where you have trees in these surrounding naturally occurring populations around these planted stands that are the same species as the species that you planted. And so they will have contributed seed and seedlings. This is natural regeneration. So what this essentially is showing us is that the stands that we're harvesting are probably a mix of the seed that we planted as well as seed coming from natural populations surrounding it. Which means that when we're looking at genetic diversity of what's actually on the landscape, it's actually increasing most likely from our seed lots. And so they're getting this nice buffer from our natural stands. And so overall, the genetic research that we had done here showed that not only are we meeting the policy minimum, but we actually probably have higher genetic diversity going on in the landscape than that policy would imply. So we're going to switch tacks here a bit and talk about managing genetic diversity for conservation. And so when we're looking at commercial forestry, we're focusing on just a few species, but we know that there are many different native tree species that exist within the province. And so this was tackled with a collaboration between MOF and UBC, where researchers at UBC did a gap analysis assessing the conservation status of our native tree species in BC. And so they looked across all 43 native species. We have 18 broadleaves as well as 25 conifers. What they were looking at was ensuring that we are capturing genetic variation across the species range in BC by ensuring there were a sufficiently large population size being conserved, so targeting that an NE of 1,000, which based on that graph we saw previously is capturing all of the heterozygosity and ensuring it's sticking around for multiple generations, as well as ensuring there's a sufficient number of populations. And so if we're looking at capturing genetic variation, the ideal way to look at this would be to have genetic data from across a species range. But this is very rarely available. So we have some genomic resources for our commercial forestry species and a few other of the native species within BC. But a lot of species have no genomic resources. They don't have any genetic markers developed. And even if they do, it's still really expensive to get genetic data. And so instead, MOF and the UBC study used BEC or biogeoclimatic zones of BC as a proxy. And so the idea here is that we know that trees have local adaptation to different climates and environments, which would correspond to different BEC zones. And so by ensuring that sufficient populations are conserved within each BEC zone, that should also mean that you're conserving that full range of genetic diversity. And so conservation for these species can either be done in situ, which means you're conserving the species inside its natural habitat. And so one way for this is protected areas such as parks. And so the rule that was established in this study was that there had to be at least 3 protected areas within each BEC zone that species is found in BC, and that they had to be at least 10 hectares each. So that's getting at both conserving the right number of populations and also of that sufficient size. So you're getting the target NE. And if there were not 3 protected areas or they weren't a sufficient size, then we move to ex situ conservation, which is the conservation of a species outside of its habitat. And so here in BC, that's done using the BC Tree Seed Center, which is located in Surrey. And so someone will actually go out to these natural populations located in these different BEC zones and collect the seed, which is then stored within the seed center. And so the scientists there also ensure that this seed is viable moving into the future. And so it's consistently checked to ensure that we're not just throwing seed into storage and assuming that we'll be able to plant it years down the line. And so if this is something of interest to you, all of this data is publicly available. So you can go to the Climate BC site, which is maintained by Tongli Wang from UBC. And there is a tab there for the conservation status of native tree species in BC. And so you can look at each species and it will show you which Beck zones it exists within BC. So that's any of the non-gray cells, and tell you how well it's being conserved and how many protected areas it's being conserved across in each of those zones. Now, of course, there are special cases. There are situations where just conserving a species, whether it's in situ or ex situ, will not make sure that that species actually survives in the wild. And as I said earlier, whitebark pine is one of the perfect examples of this. So it is a keystone species within alpine ecosystems, but it has been on the decline historically due to fire suppression, which has allowed there to be a lot of competition by those species that would normally be burned in a fire. So it's essentially losing habitat, as well as climate change. Then there was the pine beetle, and now there's white pine blister rust, which is currently its biggest threat. And so this species was the first tree species listed as endangered in the US and has been listed as endangered under SARA in Canada since 2010. And so the idea here is that just conserving the habitat of whitebark pine is not going to save it because those trees will still die due to white pine blister rust, which is a fungal pathogen shown here. It's that kind of cool looking orange fungus on the bark there, but you can see that it is massively destroying these populations. And so instead, rather than just focusing on conserving habitat, MOF, in collaboration with the government in the States, as well as NGOs across both countries, are embarking on resistance breeding for this species. And so essentially, much like those common garden or provenance experiments, they're taking individuals from across populations, across whitebark's range, and planting them in these trials where they expose them to white pine blister rust, whether the exposure is natural because we know it's existing in the environment, or if they're being planted outside of their natural habitat. And so here, this picture is actually showing a farm field trial near Kelowna, where whitebark does not naturally exist. The white pine blister rust is actually artificially exposed to it using these ribes or currant gardens where the white pine blister rust will grow, and then they'll essentially just put the leaves out by the whitebark pine. And so what they'll check then is how well these individuals in different provenances react to the white pine blister rust in order to identify species that are more resistant, with the idea that these resistant species can then be used as parents in seed orchards. And so here is showing a seed orchard in Elkhart. So it's right along the highway that comes out of Kelowna. But unlike our commercial forestry species, we're hoping to have more parents within these seed orchards than you might have for a commercial species. Because unlike when we were focusing on a single generation for our commercial species, we know that any whitebark pine that are being planted for conservation, the goal is that they will persist indefinitely in the landscape. And so we're aiming more towards the right side of that graph and trying to build up well past any of 10 to have that balance of gain in resistance breeding while still having high genetic diversity. And so for those of you who are located in Smithers, there are a couple whitebark pine trials near you. They are not bringing in the rust. They're just letting these trees be naturally exposed to the rust that already exists in the environment, but where we are checking for different varying levels of resistance. And so there's one on Hudson Bay Mountain, another in Canyon Creek, another in Sweeney Lake. This is where I point out someone who has mostly done this work, who I'm now getting to work with. Charlie Cartwright is responsible for a lot of the trials that have gone in the ground in BC for whitebark pine for this resistance breeding work. Okay, so now we're looking towards the future, since we know this is what is currently in place, but we know things are changing. So when we're looking across our forest landscapes, we know that they are now undergoing climate change, as well as those associated risks like extreme drought and fungal pathogens and increased insect outbreaks. We also know that things are changing in terms of the annual allowable cut, as well as maybe how the land is being harvested and reforested, as well as changing in governance as First Nations start to take a larger role in the decisions that are being made on the landscape. And so this is just to say that yes, all of this research has been done over the past couple of decades, but MOF does know that things are on the horizon. And so we are constantly doing research to better understand how to make these forests best react and best be the most resilient to climate change moving forward, so that we can ensure that we still have this balance of gain and diversity when it's actually out on the landscape. And so that is all I have for you. So thank you all very much. Hopefully, I have not scared you off genetics forever, and I'd be happy to take questions. Or if you come up with something later or want any of those papers that I discussed, feel free to send me an email.